The appropriate folding of proteins and assembly into functional complexes are crucial to maintain cellular functionality. Christian Anfinsen received the 1972 Nobel Prize for Chemistry for his thermodynamic hypothesis, which implied that the native conformation of a protein is primarily determined by the amino acid sequence. In the crowded cellular environment, however, the concept of unassisted folding does not hold and requires to be guided by molecular chaperones. Chaperones not only control folding, but also the timely degradation of proteins when damaged or no longer needed. A family of so-called heat shock proteins form the largest group of chaperones. To meet the demand of folding of the thousands of proteins in a cell, an intricate network of HSP families exists. Our lab mainly studies so-called HSP70 machines, a central hub in the protein quality control network. And the HSP70 protein can be considered as the central engine which receives specific input and instructions for output from its co-chaperones, DNA Js, and nucleotide exchange factors. How input and output are regulated, or how the machine knows whether a protein needs to be folded or degraded, is still largely unknown. Such knowledge is, however, crucial since protein folding is not only essential for function, but incorrect folding or failed degradation can cause proteins to form aggregates or amyloids that can threaten cellular function and cause cell death. Protein aggregation, or amyloidogenesis, is a typical feature of aging and is thought to be the underlying cause of several age-related neurodegenerative diseases such as Alzheimer's, Parkinson's, and Huntington's disease, as well as cardiovascular diseases. In these diseases, specific proteins form amyloids later in life. Understanding how the chaperone networks function to handle such specific disease-associated proteins may open new avenues for selective targeting the HSP70 machine to prevent or cure a range of human diseases and decelerate the aging process. One specific example of this concerns the discovery of an HSP70 co-chaperone, DNA JB6. In a screen, we found this DNA JB6 to be a superior suppressor of aggregation of proteins with expanded polyglutamine stretches that cause Huntington's disease, spinal and bulbar muscular atrophy, and different forms of spinocerebellar ataxias. The effects of DNA JB6 also translated into cytoprotective effects in neurons and in organismal polyQ xenopus models and Drosophila models. Expression of DNA JB6 in Huntington flies led to substantial lifespan extensions, even more so in Huntington mice with DNA JB6 overexpression. Polyq aggregation and the onset of degenerative features were delayed, and lifespan was extended by over 20%. In collaboration with others, we were able to identify a stretch of amino acids only present in DNA JB6 and not in other HSP70 co-chaperones that is responsible for its effective anti-aggregation capacity. We also found that DNA JB6 has more general anti-amyloidogenic properties, which are not restricted to polyQ-containing proteins, but also include formation of amyloid fibrils generated by the amyloid beta peptide that is linked to the induction of Alzheimer's disease. DNA JB6 may also have implications for Creutzfeldt-Jakob disease, the human variant of the mad cow disease or prion disease. Independent observation by other groups showed that the DNA JB6 could also cure a number of distinct amyloidogenic prions in yeast, supporting its even wider application as target for therapy in amyloid disorders. Together, these data suggest that DNA JB6 is a key player in protecting cells from detrimental amyloid depositions. Therefore, our current research thus entails how to upregulate or reactivate DNA JB6 to prevent diseases caused by amyloidogenesis.